My name is Irene Kaplow. I'm a Lane postdoctoral fellow in the Fenning Lab at Carnegie Mellon University, and today I will be telling you about a comparative genomics approach to identifying candidate enhancers associated with phenotypes. This is joint work with Daniel Schaefer, a fantastic undergraduate in the Fenning Lab. Large brains have evolved multiple times during mammalian history. For example, humans have very large brains relative to those of many of our close relatives, where dolphins, which are much more distantly related from us, also have large brains. When you consider brain size relative to body size, you see that humans have much larger brains than you would expect based on body size, where our close relatives, the chimpanzee and the rhesus macaque, have smaller brains relative to body size. Likewise, dolphins have very large brains relative to body size, where their close relatives, the sperm blue whales, have small brains relative to body size. So how did this difference in brain size come about? A previous study suggested that differences between brain size in mammals have occurred in part due to differences in gene expression. What I mean by that is, let's say we have an enhancer that regulates the expression of a gene in the human brain, and that this gene is involved in brain size. If there is a difference in that enhancer between the human and the chimpanzee, that could cause a difference in transcription factor binding at that enhancer, which could cause a difference in gene expression of the gene it regulates in the brain, and thus a difference in brain size. The previous study investigated the gene expression evolution underlying brain size by identifying a non-coding human-specific deletion near a tumor suppressor gene, suggesting that this deletion causes this tumor suppressor gene to have lower expression, allowing human brains to grow larger. Now, this human-specific deletion, the versions in multiple other species have been shown to activate gene expression in the brain. So we wanted to know if this human-specific deletion is also deleted in the bottlenose dolphin and the harbor porpoise, which have large brains relative to body size. So we used many newer genomes to look for this region, and we found that it's actually present in all of the cetaceans we have genomes for, which are our marine mammals, showing that there must be more to the brain size evolution story. We therefore wanted to find an approach to identify additional candidate enhancers involved in the evolution of brain size. To do this, we first need to identify brain enhancers in some species using an experimental assay. We then need to use the sequences underlying those enhancers to train a machine learning model to predict brain enhancer activity. After that, we need to use that model to make predictions of brain enhancer activity at the orthologs of the enhancers we identified in many other mammals. So just to explain a little more what I mean, we initially have this matrix where each row is an enhancer and each column is a species, and we only have data for a few columns in the matrix. And we want to train our machine learning model to be able to fill in most of the rest of this matrix. So once we have these predictions across large numbers of mammals, we need to associate them with brain size. And finally, we need to compute the statistical significance of our association. In order to do this, we developed a new framework called TACIT-ML, which stands for Tissue-Aware Conservation Influence through Machine Learning. In TACIT-ML, we first collect Open Chromatin data in our tissue of interest from multiple species, and we use this Open Chromatin data as a proxy for enhancer activity. We then train a convolutional neural network, which we abbreviate as CNN, to take the sequences underlying these Open Chromatin regions and their orthologs in the other species for which we give data to predict Open Chromatin in our tissue. After that, we make predictions across many different mammals at the orthologs of these enhancers. We then associate these predictions with our phenotype of interest using a method called phylolem. And finally, we compute a p-value for this association using a type of permutation test called phylogenetic permutations. I will begin by describing the first three steps of TACIT-ML in a little more detail and how we applied them to brain size. So we generated open chromatin data from 2 tissues in each of 4 to 5 mammals. So for motor cortex, we used 4 mammals of different levels of relatedness: the house mouse, the Norway rat, the more distantly related rhesus macaque, and the even more distantly related Egyptian fruit bat. And from liver, we have 5 mammals of different levels of relatedness. Most of this open chromatin data was generated by my lab mates, Morgan Worthman, Alyssa Lawler, and Ashley Brown, and we supplemented some of the liver data with publicly available data from additional species. We then used this data to train our machine learning models to predict motor cortex and liver enhancers. For the purposes of this talk, I will be telling you about some of our results from our motor cortex models, and I'd be happy to discuss some of the results for our liver models offline. So we make predictions across large numbers of mammals using a multi-species alignment provided by the Xenomia Consortium. The Xenomia Consortium has now sequenced over 200 mammals. Specifically, it's sequenced 222 boroeutherians, which include the rodents, the primates, the cetaceans, the ungulates, and the bats, as well as all of our closest relatives, and created a multi-species alignment that allows us to trace orthologs of enhancers. So once we have these models, we then need to evaluate how well they work. And in my ISMB talk last year, which the material of which is now a preprint on bioRxiv, I described multiple new evaluation metrics for evaluating methods for cross-species enhancer prediction, including evaluating lineage-specific enhancer prediction performance, the performance on tissue-specific enhancers, and showing that as you get further away from a species in which an enhancer was identified, the predictions on average decrease. Before training a model using all of our data, I wanted to evaluate whether the regulatory code for motor cortex is actually conserved across all of these mammals with different levels of relatedness. So to do this, I trained a machine learning model to predict motor cortex enhancers in mouse and used it to make predictions in all of the species. And what I found is that the models not only work well overall, but they also work well specifically in regions within these other species whose orthologs in mouse have a different motor cortex open chromatin status, suggesting that the motor cortex regulatory code actually is highly conserved across all of these species. I then put all of the data together and trained a multi-species motor cortex model and found that the model works well overall, works well in lineage-specific enhancers, in tissue-specific enhancers, and in brain region-specific enhancers. In addition, I showed that the predictions across species approximately match what we would expect based on the phylogenetic tree. So now that our models are working well, I use them to make predictions across all of the boroeutherians from the Xenomia Consortium. And then I needed to figure out how to associate these predictions with phenotypes. So to describe how we made these associations, I'll describe the last 2 steps of TACIT-ML. So what we wanted to do was find associations that cannot be explained fully by the phylogenetic tree. What I mean by that is, let's say we have this human motor cortex enhancer and that these are the predictions we make in its orthologs. And let's say that this is a toy example of our phenotype. Now, at a first glance, this looks like a very strong association. However, these predictions are also very strongly associated with how distantly related a species is to human. So, since there are many phenotypes that differ between human and our closest relatives and more distantly related mammals, it's hard to know if this enhancer is actually associated with our phenotype of interest or is associated with one of many other phenotypes. In order to solve this problem, we used a method called phylolem, which is a modification of phylogenetic generalized least squares. Phylolem has previously been used to identify protein coding regions associated with phenotypes, but has not previously been used for enhancers or predicted enhancer activity. The idea of phylolem is to use generalized linear least squares to identify open chromatin regions associated with the phenotype, but have an expected covariance matrix that is based on the phylogenetic relationship between species. For binary phenotypes, we used a logistic version of phylolem. In order to compute our p-values, we used a type of permutation test called phylogenetic permutations that was published very recently. And the idea of this is to shuffle our phenotypes when making permutations, but do it in a way that preserves the general trait topology in the phylogenetic tree. This worked very well for continuous traits, but it was slower for binary traits. So Daniel implemented a speedup for binary traits that decreased the runtime by about an order of magnitude. Using this, Daniel was able to find 34 motor cortex enhancers associated with the evolution of brain size relative to body size. 8 of these enhancers are within 1 megabase of genes whose mutations are associated with microcephaly and macrocephaly, suggesting that they may truly be directly related to brain size. 18 enhancers are within 1 megabase of genes whose mutations are associated with neurodevelopmental disorders, and 5 enhancers are within 1 megabase of genes that have been implicated in the growth or spread of brain cancer. So now I'm going to zoom in on one of these enhancers, and here you can see the predictions in cetaceans. So the x-axis is the predicted motor cortex enhancer activity in our cetaceans, and the y-axis is the brain size residual. And you can see in the cetaceans, we have this very strong negative association of predicted motor cortex enhancer activity and brain size. So to zoom in on this a little more, we can look at the exact predictions for each of our cetaceans and compare those to the brain size residual. Specifically, if we look at this pair where we have the killer whale with a small brain size residual and the bottlenose dolphin with a very large brain size residual, we see that the killer whale has much higher predicted enhancer activity. Likewise, if we look at this pair of closely related species, where we have the beluga whale with a small brain size residual and the harbor porpoise with a large brain size residual, we see that the beluga whale has much larger predicted enhancer activity. Finally, if we look at this pair of minke whales, where both have small brain size relative to body size, and one is actually much smaller than expected relative to the other, we see that the one with a smaller brain size relative to body size has more confidently predicted motor cortex enhancer activity. So Daniel investigated then where this motor cortex enhancer is in the genome. So here you can see a browser shot of this minke whale. And what we see is that this enhancer is near the gene LRIG1. So LRIG1 is highly expressed in the mouse cerebral cortex, and mice have small brains. In addition, there's a previous study that showed that LRIG1 regulates quiescence in adult neurostem cells, and suggesting specifically that higher expression of LRIG1 is associated with more quiescence, which is consistent with this negative association of enhancer activity and brain size. In addition, a paper showed that LRIG1 is associated with neurostem cell proliferation during development. Now, I should mention there are other open chromatin region orthologs near LRIG1, but none of them have predicted enhancer activity across species associated with brain size. We also found another motor cortex enhancer whose prediction activity is associated with brain size in the great apes. So here we see that the humans, which have very large brain size residuals, have very low predicted motor cortex enhancer activity, where these other great apes that have smaller brain size residuals have high predicted motor cortex enhancer activity. So Daniel looked into where this enhancer is and found that it's near the genes TAF1D, MED17, and VSTM5. MED17, VSTM5, and TAF1D are expressed in mouse cerebral cortex, and mutations in MED17 are associated with microcephaly and other neurodevelopmental disorders. In addition, VSTM5 regulates neuronal morphology and migration. So we wanted to know why this open chromatin region is predicted to be active in the chimp, but not in the human. So we looked at the exact sequence of this in the chimp, and specifically, we interpreted our motor cortex models and looked for the motifs found by our interpretation. And here is an example of one motif match that we found. This is similar to the motif for the transcription factor CTCF, which was found when we interpreted our models. And if we look at the human ortholog of this region, we see that it has an A in this position instead of a G, and this G is very non-degenerate in the CTCF motif. This suggests that it's possible that this enhancer is active in chimpanzee but not in human because CTCF may be unable to bind the human ortholog. Today, I have shown you how we use TACIT-ML to identify motor cortex enhancers associated with brain size. We have now done this for other phenotypes as well as for liver enhancers, and actually very recently we've done this for specific brain cell types and some of these phenotypes. In conclusion, we have developed TACIT-ML, which associates predicted enhancer activity of enhancer orthologs with phenotypes. We use TACIT-ML to identify motor cortex enhancers associated with brain size. And while today I showed you how to use TACIT-ML for a specific tissue and phenotype, TACIT-ML can be applied to any phenotype with annotations in dozens of species and Open Chromatin data and a relevant tissue or cell type from at least a few species. I would like to thank my postdoc advisor, Andreas Fenning, Daniel Schaefer, the undergraduate who worked with me on a lot of this project, Morgan Worthland, Alyssa Lawler, and Ashley Brown, who generated a lot of the Open Chromatin data we used, as well as multiple other members of the Fenning Lab involved in this project, our funding sources, and our many wonderful collaborators. I would be happy to take any questions.